15 minutes.
Hour and 15, okay, good. Okay, so I guess like many of the other lecturers, I've been given 4 time slots over 2 days. And I want to talk about a variety of issues in the physics of biological systems. There's going to be 2 themes. Today, the theme is going to be about examples of where biological systems exhibit extraordinarily precise behaviors. And that precision is particularly challenging if you think about it from the point of view of physics. And I think that that is a path into developing theories for how these systems work. And then tomorrow, the theme will be emergent behaviors in the spirit that you've heard already, focusing in particular on neural systems. I want to take, at the risk of it going on for too long, I want to take a few minutes since this is, after all, a school that's supposed to be preparatory for a statistical physics meeting, I want to take a few minutes to situate the development of our thinking about the physics of biological systems in a little bit of history. You've probably heard some of bits and pieces of this, maybe from different points of view from other lecturers already. But hopefully there'll be something new. And to discuss the relationship between the sort of general goals of statistical physics and the particular problems of thinking about the physics of biological systems. So, it's very important not to make, in this kind of science, a mistake which we all make in our views of our own moment in history. And that is to think that our moment in history is extremely special. Right? We have limited view. I mean, this extends, right, if you ask sports fans for the greatest players in their sport, the distribution will be remarkably skewed toward people who have been active in the last 10 years. And this sort of, somehow a priori you know this can't be the right answer, but it's very hard to break away from those prejudices. So it is true that there is something about this moment in the interaction between physics and biology which is interesting and exciting, and I hope to convince you of that. But it's also true that the notion of physics and biology interacting with each other, and indeed this larger effort at interdisciplinary science, this is actually very old. So if you go back to the 19th century, you have figures whose names you know. I could keep going, who were giants of what we now refer to as classical physics, but who routinely crossed boundaries that we would now distinguish between physics, chemistry, biology, and even psychology. So I don't know what's the best, I mean, I don't want to give a whole lecture about this, but let me give you a couple of anecdotes. In acoustics, in the study of hearing, there is something called Ohm's law. It's the same Ohm, but a different law. And it is the fact that we don't hear the phase relationships between the different components of a complex sound. Okay? That it is, roughly speaking, the statement that we're phase deaf. It's also not exactly true, but like Ohm's law, right? But maybe as a marker that there was some crossover between what we would call, let's say, the psychology of perception and the physics of materials, the fact that there are Ohm's laws with the same Ohm in both cases is a sign, right? Helmholtz, of course, was the most impressive practitioner of what we would now call interdisciplinary science. I mean, you know about his contributions to thermodynamics, to waves, and so on. You may or may not know about his contributions to the study of vision. His books on the optics of the eye are still read by ophthalmology students. There is the magnificent book on the sensations of tone, which was his effort to understand our auditory perception, in particular our perception of frequency differences. If you read that book, you realize that it's not only an attempt to push from the propagation of sound waves in the air to the mechanics of the inner ear and how that influences our perception of sound. It is on the sensations of tone as the foundation for a theory of harmony. It's a big book, and if you get to the end, you find his discussion of starting with the physics of sound and the physics of the mechanics of the inner ear, how do we eventually get to the question of why we find some sounds pleasing and others not? When he became professor late in his life at a different institution, he gave an inaugural lecture which was on the unification of the sciences. And so you might think that the sciences he was talking about were physics on the one hand and some sort of physiology or biology on the other, but that's not true. In his view, that unification was already sort of old-fashioned and a very bad predictor of what would happen in the next century, but never mind. The unification that he wanted was between the natural sciences on one hand and our notions of aesthetics on the other. So he thought that if we really understood sound and our perception of sound, light and our perception of light, we would eventually understand why we find some things beautiful and other things not. So you are free to believe that this was a bridge too far, but it's clear that the idea of crossing disciplinary boundaries is not something which emerged suddenly in the late 20th century with no precursors. In fact, the attempts to make quantitative measurements on our perception of the world, whether it was our visual perception or our auditory perception, or for that matter, our tactile perception. Olfactory perception, our sense of smell, is much more difficult because it's hard to control the stimuli. But certainly, the quantitative investigation of vision and hearing in the 20th century is a subject that came to be called psychophysics, for the merger between psychology and physics. And its origins are in work in this era in the late 19th century by physicists who were precisely concerned about our perception of the world because, after all, those are the physical instruments with which we make measurements on the world around us. And as physicists, we should be worried about that. Okay? So there's a long history that goes all the way back. Okay? In the 20th century, for most of the 20th century, this is, of course, where our modern notions of disciplines emerge more fully. It's not clear that one should speak about Helmholtz crossing the boundary between physics and biology because it's not clear what that boundary was, whether those disciplinary titles existed, right? So let's say, looking back, he crossed boundaries that we would draw today, but he obviously did not. In the 20th century, as the disciplines separated, there came to be a view of the relationship between physics and biology, which is very widely repeated well into the late 20th century, so into my time as a student and young faculty member, which I don't think is actually fair to what was going on, but it was a very common view, and it was the view which, as you just heard, Anderson criticized, which was that any place where physics touched biology, it was the application of physics to biology. And in particular, it was a kind of applied physics and there was a special focus on experimental methods. So if you read the history, particularly as told by the biologists, you will learn that, for instance, the introduction of X-ray diffraction, which allowed you to study the structure of biological molecules, was incredibly important in the origins of our modern molecular view of biology, and indeed the subject of molecular biology. And so the cartoon is, well, that's what the physicists contributed. They brought, biologists had well-defined questions about the world, and physicists had tools for answering these questions. So I think this is really unfair to what actually happened and is a kind of revisionist history. Because it's also true, perhaps because the physicists had tools with which to answer questions, they asked different questions. So there's something which doesn't fit the model of applied physics and experimental methods, which is that you ask new questions. In fact, if you think about the history of the subject that we now call molecular biology, of course, physicists, people who came from the physics community, had an enormous impact. So Max Delbruck, Francis Crick, Seymour Benzer, one can go on down the list. And what many of these people did was to ask questions which the biologists had not asked. Faced with the same phenomena, they asked different questions. In particular, I think it's worth remembering that there were lots of people in the world who knew how to do X-ray diffraction to solve the structures of molecules. And indeed, that knowledge had diffused into the chemistry and biology communities. But it is not the case that biologists had articulated, even biochemists had articulated the idea that if we knew the structure of biological molecules, then we would understand how they worked. That idea was actually articulated by the people who did the X-ray diffraction experiments, who were largely physicists by training. So the idea that you should worry about the structure of biological molecules, which by now is so commonplace that it hardly needs to be mentioned, right? Everybody knows that if you want to understand how molecules work, you should look at their structure. That wasn't something people were asking in the years immediately after World War II. And fortunately, there were these physicists who knew how to do the experiment, and so they were able to come with the answer. The only people who thought to ask that were a handful of physicists and extremely physical chemists. Okay? But they also asked other questions, even earlier. So many of you probably know this famous set of lectures by Schrödinger during the Second World War called What is Life? It's a beautiful little book. It is credited with inspiring many of the people who went from a physics background to become what we would now call molecular biologists. But that book itself is inspired by observations that were made by three people, and I can never remember how to pronounce all the names, but Max Delbruck was one of them, right? So this is a famous three-man paper. And what they do is they take the observation which had been known for many, many years, well, as many years as you could, given when X-rays were discovered. So you knew that if you exposed animals to X-rays, you would get mutations, and then those mutations would propagate and follow the Mendelian laws. Okay? And indeed, this is a practical way. We don't do it so much anymore. It's easier to feed animals nasty chemicals that cause mutations. Which we seem to know much more about today than we used to, but never mind. So it was known that you could induce mutations, and there were these abstract things called genes, and they obeyed these probabilistic rules of inheritance from one animal to another, but nobody knew what they were. This is before we knew that DNA was what carried the genetic information, and certainly before we knew what the structure of DNA was. So this is the 1930s. So what did these guys do? They measured the cross-section for producing a mutation. They showed that the probability of generating a mutation was proportional to the intensity of the X-rays. So that's also not obvious, right? It means that if you think about the about the physics of radiation interacting with matter, if you get something that's proportional to the intensity, it means that you're observing things that happen one photon at a time. Because, for example, if you needed to absorb 10 photons, then it would be proportional to the intensity to the 10th power, unless you did it coherently, which would be really weird given the low intensities of the sources. But if it's proportional, then the coefficient, is a cross-section. And it tells you, in a sense, how big is the target that you're hitting when you cause a mutation. And the answer is something of molecular dimensions. So this was the understanding. And so you should think about this. It means, again, now this is all commonplace, right? But what it means is that a single molecular event, the absorption of one X-ray photon, which is happening in, the thing which is doing the absorbing is the size of a single molecule. So a single molecular event does something which you can see macroscopically in, the color of the eyes of a fly 3 generations down the line. Okay? And so that is the inspiration for Schrödinger's discussion of the precision of transmission of genetic information and so on. And it's a question, I think, that nobody had thought to ask, right? So yes, it's true that you couldn't do this unless you knew how to manipulate the X-rays, you knew how to measure a cross-section and so on. But I think that to say that they made progress and contributed something to our understanding of the living world because they had this tool available to them really undersells the conceptual leap forward that they made when they asked, how big is the physical object that carries our genetic inheritance? Okay. An important part of this period in which the focus was on the application of physics was that the flow was, if you were a physicist and then you acquired an interest in the phenomena of life, then you became a biologist. Okay? And I mean, this is a sociological statement, right? You would have an impact on the biology community. The physics community would be very proud of you because, of course, it illustrates, as you were saying, this tendency of physicists to go into other fields. Of course, we're very proud, right? We're all so smart. We got things done when other people didn't. But you stopped being a physicist. And this really lasted a long time. And so by the time, so certainly in the as we entered the last quarter of the 20th century, this was still true. And then somewhere, bracketing 2000, and exactly where, let's say, 2000 plus or minus 15 years, which almost takes us to the present, you start to see that maybe you stay a physicist. That is to say, you start to see life as part of physics. So the phenomena that you see in the living world can be thought of as challenges to our understanding of physics rather than just opportunities to apply physics that we understand to another field. Okay? And so that's where we are today, but you have to ask, well, what changed? So I'll remind you of Max Planck's great remark about how theories succeed, that new theories succeed not because they account for the data or they're confirmed by experiment. They succeed because the proponents of old theories die. So some of this is just a generational shift, right? I mean, where the boundaries of subjects are is something that's agreed upon. It's emergent, right, as a sociological phenomenon, right? And so a different generation can decide to draw the boundaries in different places. So that's part of the answer. But I think there's a deeper answer. And when I talk to people who are, my professor's generation or my parents' generation, there were physicists who certainly were very intrigued by the phenomena of biology. But there was a sense that you couldn't really make progress in the way that we're used to in physics. So you could get excited about something and open up its exploration, but you would be changing the way biologists did things. You wouldn't really be able to do physics. So if you were honest with yourself and asked yourself the question, is it physics? There's some standard in your mind for what you're supposed to do when you do physics, and you'd have to say, no, I'm not meeting that standard. It's not that I'm forbidden from being interested in this because it's not physics. It's that I can't do science in the way that I'm used to. And this had two origins. One is that the living world is enormously much more complex than the problems that we have traditionally dealt with in physics. And so it took time for us to understand that complexity could be tamed in any context. But there was something, let's say, extremely widespread, very hard to put your finger on, but unanimous. And that was the belief that it was all just a mess. So physicists would look at what biologists did and they would say, yeah, but they don't measure numbers. Imagine you're a theoretical physicist, you would like to do something. How do we know that our theories of the natural world are true? The answer is, we predict numbers, people go out and measure them, you get them right, you get them wrong. That's it, right? That's how the field progresses. And as you know, in some areas, which are presumably not a model for our understanding of the living world, this has gone, to the 13th decimal place, which is extraordinary, right? The idea that you can both measure and calculate something out to 13 decimal places, there's only one such thing, of course, anomalous moment of the electron, but the idea that you can do that is extraordinary, okay? And it is that, and in every area of physics, we see the evolution from a qualitatively striking phenomenon to a theory to a quantitative test of those theories. In some cases, the discovery is literally a numerical discovery. So if you think, for example, about the quantization, or even more so, the fractional quantization of the Hall effect, right? It's the fact that in this messy real material, out pops a pure number multiplied by fundamental constants, right? That was a surprise. So this interaction between, and then, what does it mean to have a theory? You have to explain why you get integers out of this mess, or ratios of integers. So in every part of physics, our understanding rests on this comparison between theory and experiment, which is a detailed numerical comparison. Different levels of detail for different subfields at different moments in their history. But there's always the prospect of that numerical comparison. And if you take a random area of biology, particularly 20 years ago, and you look at its practice, pick up a journal, or go to a talk, right? Walk across your university campus to the other institute and listen to what people are doing in talks. You may hear marvelously fascinating things about the world, but you don't see, plots of data where you would say, oh, right, I need a theory because, there's this striking quantitative relationship that needs an explanation. In fact, when you push on quantitative relationships, people get soft. They say, well, it's biology, it's complicated, it's hard to control. Maybe it's not only it's hard for us to control, maybe it's even hard for the organism to control. So you have the idea that nothing in biology is particularly precise because it can't be. And so then the whole program of having quantitative theories kind of goes out the window. So what's changed, the thing that has changed in the last 20 years is not just the courage of the physics community to explore, And there's reasons for that courage. It's not just the sociology of the physics community being willing to accept these explorations as part of physics rather than as an application to the outside. What's changed is not only the quantity, but the quality of the data. So it is now possible to do physics experiments on real, live, intact biological systems with the kind of precision that we're used to, okay, maybe not in atomic physics, but certainly in condensed matter physics. And that's really important because that wasn't true, certainly when I started in this business. When I started, there was a handful of biological systems in which you could do those kinds of experiments. And so if you wanted to make a comparison between theory and experiment, you better start thinking about 1 out of 5 possible systems. And so the whole rest of the biological world was inaccessible to you, except in qualitative ways. So I think that if you're interested in the physics of biological systems, even if you're a theorist, as I am, you should care about this because it is the quality of the data that ultimately is going to make it possible for theories to really succeed. So if you have a theory which is somehow leads to qualitative explanations of the phenomena, that can be very important, particularly for phenomena that were mysterious. But in the end, you'll know that you're right by the standards of the physics community, not even by the standards of the biology community. You'll know you're right because you can make quantitative comparisons with experiment. And so that's something you really need to worry about. And it's still the case that different biological systems are in different stages of development with respect to the quality of the data. And so it behooves you either, as a theorist, either to make friends with an experimentalist who's willing to make the effort of taming some new biological system to the point where you can answer the kinds of questions that you want it to answer, or going to an example which is already well developed. And you're interested in that example not because you're interested in this particular organism or this particular biological process, but because that's the place you believe that there's something more general underneath, and that's the place where you think you have the best chance of connecting theory and experiment, okay? So that's already probably too much philosophical preface, but maybe I should stop for a second in case people have, I mean, these are sociological issues. As I say, there's a very substantive issue underneath them, but if you're worried about something, this is a good time to ask. Oh. Yes, thank you.
So you think that this kind of interaction of physics and biology has an important role in biology to step from being just observational to predict and control things? Like
So look, there's two. Okay, this is a little dangerous. I usually only do this in my office when students come to talk to me, but I'll do it in public. This goes on the web, right? What the hell? So this axis is meant to be the extent to which quantitative thinking is an essential part of what we do. So I would say in physics, being quantitative is not some decoration that we add to our qualitative understanding, right? It's intrinsic. Our theories come in mathematical form. We compare with quantitative experiments. There's just no other way to do it, right? I mean, I don't want to know that the universe expands instead of contracting, right? I want numbers, okay? So it's just, it's sort of baked into the practice of our science. In biology today, in most of the science, it's qualitative description and enumeration, cataloging, and quantitative things are added on top, okay? That's not true in all fields, but in most of it. So if this is an axis of how quantitative we are, you could say, well, draw the boundary for me. What does the boundary look like between things that are biology and or the frontier of our understanding? So, a lot of things are stuck down here. There are a few things where we've been able to get out pretty far. There's a few funny places where for some reason somebody was able to land and do something very quantitative very far from everything else, right? It's not clear that they have to have simple geometry, okay? So now, as a new student, what do you want to do? So one idea is that, look, I'm a physicist. I want to go someplace where I already feel comfortable. So you go here, right? And you try to push the front outward a little bit in that direction, right? So if you want, you could say this is where the frontier of the physics of biological systems is. That's actually not a complete view. There's another view which says, maybe a frontier problem is that the community of physicists who pushed very far out in this direction, which has something to do with immunology, and the community that pushed very far out in this direction, which has something to do with sensory perception, maybe, actually, if you look closely, you notice that some of their theoretical language is common. So in the spirit of things that we often do in physics, we want to achieve unification, right? So we should look for connection, look at very well-developed areas and try and find connections. Okay, I could keep going on, but you get the spirit out here. But there's another thing you could do, which is you could work on taking this enormous area of biology and pushing it a little bit forward to become a bit more quantitative. And it's clear that this is in the same direction as the things that are out here. But in some way, you would say this is the physics of biological systems, and this is the attempt to build a more quantitative biology. So which one do you want to do? Complete matter of taste. And luck, right? If you find an opportunity to do this in a really interesting way, and all the opportunities around you to do this somehow look boring to you, you should do this, right? Because you're more interested. I would also say that by now, almost all the people who sort of think carefully about this in the biological community understand that this is necessary for their science to progress. They may not have any clear vision at all of what's out here, but this they know for sure. Sometimes they perhaps are more certain about how to do this than they should be, given what they know, but we all make that mistake from time to time. I would say, for me, my interests are out here, but there's a very important component here, which is education. So this is different in different countries, but in the United States, I will tell you that biology is sold to university students as the science you can do even if you don't like math. So on the one hand, you have the leaders of the biology community giving us lectures about how we need to build a more quantitative biology and that's the future and we're going to have personalized medicine and everything else. And on the other hand, we have people who write the curriculum for biology majors saying, do they really need to know calculus? So I think that one place where we should put aside our sociological prejudices and say, look, these goals are, and by the way, so to finish the thought, right, it's clear that what many of us would like to do out here would be much easier the further this foundation advances, right? So we really have an interest, not just as human beings who would like to benefit from personalized medicine or whatever, but as scientists in seeing this frontier advance down here, okay? And so we can put some of our energy into working together with our biological colleagues to build better educational structures that make this advance more quickly. So maybe that's an answer. Sure. Okay. So let's talk about the science. So I've been fascinated all my life, all my adult life, about the fact that biological systems, contrary to your impression that they're a bit of a mess, can be extraordinarily precise. And the classic example of this, which I'm not going to spend a lot of time talking about, but just to remind you, so we want to talk about precision of biological function. Oh, sorry. Let me make one more sort of philosophical remark about this evolution of the interaction in physics and biology. There's an intermediate period somewhere in here, or let's say near the beginning of this period, where this notion of building experimental tools to answer the biologist's question reaches a kind of zenith, where the tools become so sophisticated and what you can see becomes so spectacular that the physics community sort of wakes up and says, no, 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 okay, this really is physics. I don't care whether they're making their measurements on biological systems or not. This must be physics. And this was the advent of single molecule experiments. So the idea, for instance, that you could do an experiment where you're watching the RNA polymerase molecule move along DNA and spit out the messenger RNA, which is the transcript, right? Reading out the information that's encoded in the genome in order to eventually make proteins. The first step is you make messenger RNA. By using the optical trapping methods that have their origins in the work by Art Ashkin that was finally recognized with the Nobel Prize last year, it became possible to hold on to a single molecule of DNA, get the RNA polymerase molecule attached to a little bead, hold onto the bead with another optical trap, and watch it as it walks along. And you could do this with a resolution that was sufficient to see it take steps base pair by base pair. The base pairs along DNA are separated by 3.4 angstroms. So you actually are seeing stepping motion on the scale of 3 angstroms, which means the noise level in your measurement had better be about 10 times less than that. So this isn't LIGO yet, but it fits on a desktop, so it's not so bad. Right? So you're fractions of an angstrom noise level. And you can not only see that, but if you change the chemical environment so that the right nucleotides are not present, you can see the RNA polymerase pause, right? It's moving forward and spitting things out, and suddenly it pauses and backs up. And the reason, of course, is that it's made a mistake in its proofreading, which is another place where ideas from physics had an enormous impact on our understanding of the living world. So, if you walk into a lab where they do this, it looks exactly like the atomic physics lab next door, right? Optical table, lots of lasers, incredible stabilization, everything else, because you're doing this incredibly precise measurement. And so I think that was something that the physics community could see was physics, right? Because the experiments were just so hard. And they really are very beautiful. What's different about the examples that I'm going to talk to you about today, and I think the examples that animate many of us in the community now, is that they are not that work, right, is in a reductionist mode, to use the dichotomy that Maxi was talking about this morning, right? You have a reductionist mode, but you have a synthetic mode, right? So on the one hand, we know that the phenomenon of life is built out of parts. And knowing what the parts are is obviously important and useful. So let's start taking it apart, right? In the same way that we took apart matter into molecules and atoms and electrons and nuclei. Let's do that exercise, but for the living world, and see whether there are components which are unique and different. And there are, obviously not the atoms, right? But at an intermediate scale, the molecules are characteristic of life, and they're universal across the living world. So in that sense, it's true that biologists are very fond of diversity, but the molecular biologists, perhaps driven by the fact that many of the first ones were physicists, the subject of molecular biology exists because the molecular components have a degree of universality, right? I can take your DNA and I can copy it using the DNA polymerase that's taken out of a bacterium that lives in the vents at the bottom of the trenches in the ocean, right? And at no point in evolutionary history, at no point in evolutionary history did we ever encounter each other. But I take the DNA polymerase out of that bacterium, and I can use it to copy your and my DNA. Okay? So there is a universality in biology at the molecular level. What animates many of us today is something that connects more with the synthetic part of physics, because we're interested in how the whole system functions. We're not just interested in the parts. And so all the examples I'm going to give you are at least anchored by observations about function of the intact system. And that also carries with it the challenge of doing a physics experiment. Again, I'm a theorist, but I really appreciate what my experimental colleagues do. It's one thing to take a single molecule out and put it in a box and say, I can do an experiment on this one molecule, and it looks like a physics experiment, and it's that precise. Sure, all the molecules are the same, and it's like doing, it's not easy in the same sense that it's not easy to put a single electron in a box. But you understand that if you put a single molecule in a box, if you do it again tomorrow, you get the same answer. If I put a single organism in a box, do I get the same answer when I do it again tomorrow? That's a much harder problem, okay? So the idea that we can do physics-level experiments on cells, organisms, groups of organisms, and so on, there's something deep there that we've learned how to do. So let me talk about the precision of biological function. The classic example is the ability of our visual system to count single photons. So if I put you in a dark room and I let you sit there for half an hour or so you're completely dark adapted, and then I flash a dim light, and I ask you, what's the probability that you saw it? The answer is, of course, that if the flash is very dim, you don't see it, ever. And if the flash is very bright, you see it every time. The question is, what's happening in between? And in a saga that begins with a suggestion by Lorentz in 1911, and has an important peak in the 1940s with experiments both in Europe and in the United States. You may remember that there were other things going on in the 1940s, so life was complicated. You can measure this curve with reasonably high accuracy. And there's a very simple theory that explains this, which is that the probability that your retina, that n photons will arrive at your retina, given the intensity of the flash, is a Poisson distribution for any normal light source. And seeing is if you count more than a certain threshold number of photons. And in the early experiments, this number is about 6. And you realize that this theory has two parameters, k and this efficiency alpha. If you plot versus the logarithm of the intensity of the flash, then your uncertainty about alpha just corresponds to translating the curve rigidly. And so the shape of this curve is determined entirely by k. And so you can look at observer after observer and ask yourself whether they have the same value of k, and the answer is yes, and it's 6. But in those experiments, the flash of light is being delivered to an area of the retina that has hundreds of photoreceptors in it. And so that means that the probability of any single photoreceptor receiving 2 photons is negligible. And so it must mean that each photoreceptor responds to one photon. And more importantly, that single photon event contributes to your perception. And there's a whole series of other things that surround this. For example, if you're willing to say, yes, I saw it when you counted 6 photons, why do you need to count up to 6? And the answer is that, as in any photodetector, you expect a certain amount of dark noise. And so the question of whether you need to count up to 6 is determined by how severely I punish you for false alarms. And it turns out that at the beginning, actually, the 2 different groups of this experiment did that very differently. One of them was very strict about false alarms, and they found k equals 6. The other one was much looser. They found k equals 2. And after a few years, people realized the difference must be must be the result of the dark noise. This gives you an estimate for what the dark noise is. Okay. All of this is being done on human observers. It would take until 1979. So it starts with Lorentz in 1911. These experiments are from the 1940s. By 1979, you get electrical signals from single photoreceptors. And you can directly observe the single photon responses. Now you can, of course, check that they really are single photon responses, not just that they have false-alarm statistics, but you can measure the cross-section for producing them and all that. And you even see that when the receptor cell is sitting there in the dark with some frequency at random, with some rate, probability per unit time at random, it produces a little pulse that looks exactly like a single photon response. And the rate of those pulses is exactly what was needed to explain why you need to count up to 6. And by the way, if you cool the system down, the rate of these spontaneous events, of course, goes down because it's a thermal noise source. In fact, it's a spontaneous change in the structure of the molecule that's absorbing the light. So that means that if I cooled you down, I would lower the dark noise level, which, of course, our friends who do microscopy and especially astronomy all know very well. No picture that you have seen of the universe recently was collected from a camera whose detector is at room temperature, right? They're all being cooled. Of course, for us, this isn't an option because we're warm-blooded. So we're working very hard to keep everything at 37 degrees centigrade. But if you're a frog, that's not true. If I cool off the frog, then the noise level will go down. And so he should be able to see dimmer lights because he doesn't need to count up to such a high threshold. Except, of course, if you cool him off too much, he stops moving altogether. And the prediction of how much better he should see works all the way down to the point where he stops moving. Okay. So this is a fantastic chapter in the physics of biological systems, and it rests on the observation that the system is capable of performing at the limit of what physics allows you to do. You count every single photon, and in the end, you're limited by the dark noise in the receptors that you use in the same way that our astronomer friends are limited by the dark noise in the detectors that they use as they try to observe the universe. It's a beautiful subject. Okay. You might worry that this is somehow a very small corner of the biological world. By the way, it's important to realize this isn't only about what the receptor cells can do, right? It's about what the rest of your brain can do. Because these signals are being taught, they go through the retina and they get tossed into your brain, which again, if you stick electrodes into brains, you see all sorts of noisy, messy stuff. And yet somehow these single photon signals make it all the way through to the point where somebody raises their hand and says, yes, I saw it. Okay? And by the way, we don't understand how that works, just to be clear. Okay, we understand a lot of what happens as the signals pass through the retina, but once they go up into the brain as a whole, this is not a part of vision that's been very well studied in the central part of the nervous system. So this is a corner. It's vision. It's about your brain, and it's vision in when it's dark outside, right? This is where it matters. So it's in some corner of biological function. What about something that's more central to biology? So this is one example. What I'm going to do, instead of giving you a long catalog of lots of examples, I'll give you this one, which was the sort of inspiring one, and then I'll spend the rest of the time focused on one class of problems. And this is a class of problems where the signals are not being carried by light or by sound, they're being carried by changes in the concentration of molecules. And this is generic in biology. All over, you have places where the concentration of molecules outside the cell influences what happens inside the cell, changes in the concentration of molecules inside the cell influence other processes, and so on. And so if it turns out that living systems are capable of making measurements that are very precise when it comes to detecting small changes in concentration and having those influence subsequent processes, then that's something much more at the core of all of life. So where are we going to go look for an example of this? We need, there we are. We need someplace, well, yeah, half of it works. There we are. We need a place to look, which is, okay. Right, there are two of these lectures, good thing, about the same subject, so I can see how far I can get. We need a place to look where as here, right, you don't start by recording from the receptor cells. You start by looking at the behavior of the whole animal. So let's pick an example where we can start with the behavior of the whole animal. So this animal, although you can't tell by looking, is a fly. It's a fly before it becomes a fly. This is the egg of a fly, the embryo of a fly. It is 0.5 millimeter in length. And the reason that flies are a favorite object of study is, first of all, they reproduce very quickly. So if you want to do genetics, the life cycle is short. And if you want to study the development of an embryo, instead of waiting 9 months, you wait 1 day. So the mother fly lays an egg, and 1 day later, the little maggot walks away. It's pretty amazing. Okay? And remember, right, it works the same way in every organism. You lay an egg, it's one cell. It's one very big cell. And then it's going to divide into many cells. And along the way, cells are going to figure out what they're supposed to do in the fully developed organism. And so this is a moment about 3 hours after the egg has been laid, where every cell that you're looking at actually, in a sense that I hope you'll be a little clearer about before the talk is over, every cell knows what it's supposed to do. Okay? And it's about to do some of it. So what are you looking at? What you're looking at here, so these are electron micrographs, and you're looking at the animal from at the egg from the side, and here to here is 15 minutes. It's electron microscopy, right? So you have to stop everything. So this was stopped 15 minutes before this one. It's not the same one, right? And you'll notice that at this stage, all the cells look alike. And here, something has changed. There's a furrow underneath here, and there's a furrow here. So let me tell you about this one underneath first. I'm not going to say more about it, but just so that you know what's happening. If you get many cell divisions, you get a ball of cells. Now in the fly, there's a little gift to the experimentalists, which is that most of the cells have moved to the surface of the ball. There's really only a few cells inside, all of the actions happening on the surface. That's nice because then you can see it, right? The interior is quite opaque. So if you want to do microscopy, it's easier if everything happens at the surface. The egg thus starts out, or the embryo thus starts out with an outside and an inside, right? It's topologically a sphere. We, and all other organisms, are not topologically spheres. Right? We have two outside surfaces, this one and this one. And, well, we're sufficiently spaced from meals that you can think about the fact that this one goes all the way through your body, right? So the topology of the thing has to change at some point. And this is sometimes jokingly referred to as the most important moment in your life, because if you don't get this one right, that's it, okay? There are other choices you have to make in life that are also very important, but this one, there's no, there are no divorces, right? You can't go back. If you get it wrong, you're dead. So this, underneath here, this furrow is the beginning of that process. This ball of cells is crumpling in on is folding in on itself, and eventually that fold will break, right? And you'll have two outside surfaces, right? You'll have two layers of cells, one of which is the nominal outside and one of which is outside relative to the yolk inside the cell, okay? Let me focus on this one because I'm mostly interested in how patterns form along the length of the embryo. This one divides what will become the head from the rest of the body. So now, you would like to see this forming in real life. So what you do is you genetically engineer flies so that they make some protein which glows, right, which is fluorescent. So there are these famous green fluorescent proteins that were first isolated in jellyfish. There are modifications of these proteins. You can use them to tag other proteins and so on. So you genetically engineer the fly so that it makes something that glows. Here, we don't need this to be a quantitative experiment yet. It's not quantitative because the brightness of this spot means something. It's quantitative for a reason that you'll see in just a moment, which is that you can watch, that doesn't work. Okay, there's no clever way of stopping it. You watch this fold happening. So what's being done in this experiment, which is done by my colleague Thomas Greger? What you do is you look down on the embryo. You look down on the embryo with a light microscope, and you put the plane of focus at the widest part. And since all the cells are around the edge, what you see is just that edge, because the cells are glowing. And you see this fold happening as it, from this point of view, going into the board, but from this point of view, pinching in top and bottom, okay? So this lecture is supposed to be about the tremendous precision of biological systems. So what's precise about this? The answer is, if you look at 100 embryos, and you measure where does this fold happen between here and there, the answer is at 31.5% of the way from here to there, plus or minus 1%. So genetically identical flies, or as nearly genetically identical as you can make just by inbreeding in the laboratory, okay? If you do this, if you put a piece of rotten fruit outside and collect lots of fruit flies, they're enormously diverse genetically. They're going to be different sizes, everything else, okay? So let's do this. Let's do a controlled experiment. Let's basically let the same organism run the same program over and over and over again, which in this case is just the mother laying many, many eggs. And what you get is that this point is accurate to 1% of the length. If you count, you'll find fewer than 100 cells along the length. So that means that cell number 31 knows that it should eventually become part of the head, and cell number 32 knows that it should become part of the rest of the body. And you can't do better than that. So the question is, where is the signal that tells the animal to do this, tells the embryo to do this? So this is one of one of the great success stories of modern biology. In an extraordinary series of experiments in the late 1970s, Eric Wieschaus, whom I have the pleasure of collaborating with, and Jannie Nusslein-Volhart did an experiment in which they made lots of mutations in fruit flies by X-rays, by nasty chemicals, all sorts of ways. And they searched for mutations that produce defects in the body plan of the animal, some of which are visible early in embryonic development, others take longer to develop. And they whittled their way down to show that although they looked at thousands and thousands of different mutant flies, after a while, when you start crossing them with each other, you realize that some of those are just different mutations in the same gene. And when you're done, you realize that there's only about 100 genes that are needed to make the pattern that becomes the body plan. So that means that the whole problem of obviously the problem of actually moving things around, right? That takes more proteins, but you need those later in life anyway, so they're not specifically involved in embryonic development. They're just things you need for the general business of being alive. But it only takes about 100 genes to specify the body plan. That's pretty extraordinary. Flies are kind of complicated creatures, so the idea that you can build all that with the instructions that are in 100 genes is amazing. And if you focus only on the axis, the long axis, then really it's more like a couple dozen. Okay? So what do those molecules do? Well, so let me channel Eric Wieschaus for a moment. Eric, they would go on to share the Nobel Prize for this work. And Eric is very philosophical about scientific impact. And what he explained to me once was that the experiment that they did was really a very good experiment. He is not falsely modest. He said, however, the end result of that is that you have 100 different bottles with inbred flies, each of which carries the mutation in one gene that you know is relevant. What do you do with that? Well, fortunately, while they were doing this, the second wave of molecular biology happened. And it became possible to reach into an organism, pull out the gene that you were interested in, sequence it in the laboratory, make the proteins, put things back into organisms and see and manipulate those genes and so on. So genetic engineering, right, the second generation of molecular biology happened while they were doing this experiment. And so they could take the 100 bottles of flies, distribute them through 100 laboratories around the world, and within a few years, it was not only the case that we knew there were only 100 molecules that were relevant, but you had those molecules in your hand. And you could start asking what they did. And almost all of the molecules that are crucial do only one thing. They regulate whether other genes are read or out or not. So they're almost all transcription factors, which are proteins that bind to DNA and regulate the transcription into messenger RNA to whether that information is read out or not. So what that means is that the control of the body plan is determined, the form of the body plan is set by a genetic network, right? You have transcription factor proteins, which when they're synthesized, they regulate whether other transcription factor proteins are being synthesized, and there are loops, right? So quite remarkably, what this tells you is that in the first 2.5 hours of the embryo's life, all of the molecules that are really doing anything important are just turning each other on and off. And then there's a pause, and at that point, you start moving things around. So the laying down of the body plan and the building of the structure are separated from each other. Didn't have to be that way, right? And since we know what molecules there are that lay out the body plan, we can read the blueprint. So I'm fast forwarding, right? You know which molecules they are, you isolate them, you figure out what they do, da, 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 da. And now, for instance, you can measure their concentration in the embryo. And I'll tell you in a couple minutes exactly how you do that, but I want to get to a good pausing point. So if I choose, so here's 0 and here's L, the same long axis that you have up there. And if you choose the right molecule, good, and you measure its concentration, you find that it comes in stripes. Now, some of you may know, you've probably not looked in detail at what comes out of a fly's egg, but that's because they're called maggots, and they're associated in our minds with death and decay. Of course, these are fruit flies, so the only thing decaying is fruit, right? They don't actually normally live on decaying flesh, but somehow this leads us not to think that fly larvae, maggots, are so charming. So the more attractive alternative is a caterpillar, which becomes a butterfly, and that's nice, right? So, but same idea, right? They're all insects, it's all related. So if you look at a caterpillar, you surely notice that the body comes in segments, and each segment has a couple legs, right? If you came away from the talk thinking that these stripes become the segments, that wouldn't be so terrible. It's not exactly right, but it's close. Furthermore, if you look not just at the concentration of one of these molecules, but two, you see that you get interdigitating stripes. If you look very carefully, you discover that the offsets between the different stripes are 1 or 2 cell spacings, depending on exactly which molecules you look at. And if you choose the right molecules, you discover that that furrow that you're seeing is drawn here, between the first 2 stripes. Okay? So the signals that produce the body plan, that produce all the movements, are contained in the concentrations of these molecules. If you do the same experiment over here of looking at 100 embryos and asking, what is the position of this stripe and how much does it vary? The answer here is also plus or minus 1% of L. And so that tells you that the precision of these molecular signals is high enough to drive what you observe. Okay? There's no mystery. Well, there's a mystery. How do you get to that accuracy? But that accuracy is present there already. Okay, what makes these stripes? You might say, oh, this looks like a pattern formation problem, right? I have a periodic pattern. So you think about Turing and other examples in non-equilibrium physics where you see periodic patterns emerging. Not quite the right answer. And the reason is that the problem that Turing posed was the problem of how do you take a perfectly homogeneous egg and get a pattern to emerge from that. And it turns out that the egg is not homogeneous to begin with. When the egg is made, the mother places molecules at cardinal points. So for example, there is a molecule whose concentration looks like this. Okay, same scale. And the reason is that the mother has placed the messenger RNA for this molecule at the end of the embryo that will become the head. So there's a source of these molecules here. When the egg is laid, the messenger RNA begins to be translated into protein. The messenger RNA is stuck down to the end of the embryo, but the protein is not, so it can move through the embryo, roughly by diffusion. And it also decays because it's a protein and it's getting chewed up by the machinery that cells have for chewing up proteins. And so the result is you have a box, you have a source of molecules at one end, they diffuse through the box and are degraded. So what do you expect? You expect that you'll have an exponentially decaying profile of concentration if you reach steady state, and that's what you see. And if you manipulate the concentration of this molecule, you move this furrow that determines where the head is back and forth. Okay? So ultimately, the concentration of this molecule, and it turns out there's another one that goes the other way, and there's a third one that is symmetric, just for completeness. The concentrations of these molecules are the ultimate origin of information about position. These molecules are all transcription factors. Not exactly true. Sometimes they're one step away. They influence a transcription factor. And they feed into a genetic network that has multiple components that all interact with each other in some complicated way. And out comes these patterns, which are precise to 1%. Okay? So the real precision here is, in fact, not the 1%. The precision is something else. It's that if you ask yourself about two cells which are next to each other, separated in position by 1%, and you ask, what is the difference in concentration of these input signals that the system is seeing, right? So let's go to a place here where this signal has decayed and this signal has decayed. So there's basically one input to cells in this neighborhood. And I told you that cell number 31 and cell number 32 are going to do different things deterministically. But there's only one source of information they have, which is this concentration. So how different is the signal that drives that divergence? And the answer is, well, since this is an exponential decay, the fractional difference is always the same. That difference is 10%. So what this tells you is that cells are capable, at face value, cells are capable of making decisions which are distinct based on 10% differences in the concentration of these molecules. These molecules are transcription factors. So we know from lots of things in molecular biology, the typical concentration of transcription factors is measured in perhaps tens of nanomolar. Nanomolar is one of those units that as physicists we don't have much feeling for. So let me convert it for you. It's molecules per cubic micron. So you're in a regime where the concentration of these molecules is perhaps 10 per cubic micron. The nucleus of a cell, where all the action is happening, is a few microns across. So you're talking about hundreds or perhaps 1,000 molecules, and a 10% difference is enough to deterministically drive behavior in different directions. That's a very small difference. And in fact, when my colleagues and I started working on this, and this is where I'll pause, when my colleagues and I started working on this, this is what was in the textbooks, not least thanks to our colleague Eric. But if you took the numbers on this picture seriously, as you'll see, there are very grave problems. And in particular, the idea that cells could distinguish 10% differences in concentration is something that is surprising. In fact, it's so surprising that most biologists said, no, that can't really be how it works. There must be something missing from this picture. So even though this was the picture in the textbooks, when you confronted them with this numerical fact, they all said, no, 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 no. 10% differences in concentration can't really matter. There's another reason why you should be suspicious about 10% differences in concentration. If this concentration is what's driving cells to know where they are, ultimately driving cells to know where they are as it's processed through this network, then not only have you seen that you need two cells that see a difference of 10% to do different things, but there's another problem, which is that in two different embryos, if you want cell number 31 to do the same thing, then it had better be true that the concentration is the same to within 10%. And nobody believed that if I took two embryos and measured the absolute concentrations of signaling molecules, they would be the same to within 10%. Because that would require that the mother set everything up to 10% precision, and that all the machinery, after all, she puts in messenger RNA molecules, all the machinery for translating from messenger RNA into protein would have to run with the same efficiency in the two different embryos, all to 10% accuracy or better. Okay, so what I'm going to tell you about in the second lecture is, yes, this is true. There's a physical limit to how well you can do, which is that wherever it is that these molecules act, they act by arriving at some point, and they arrive at random, so there's a shot noise. This is very close to that shot noise limit, and in fact, presumably it's not beyond the limit, it's not smaller than the limit, but if you're not careful, you might think it is. And that's the beginning of a theory of what these networks should be doing in order to squeeze as much information as possible out of this limited number of molecules. And I'll give you our latest sort of signpost in the development of that theory. We should take a coffee break.
Questions?
Yes.
To be, there's influence in the expression of things.
Obviously, in order to do that, you have to think about mechanics, right?
Yes.
However, it's also true that I can take this picture at this stage where nobody's moved yet. So that means that it didn't have to be this way, but there is a phase of this where it's all about setting up a pattern of concentration as a function of position. And nothing moves. There's no mechanics yet. And then things start to move and everything's coupled to everything else, and I agree with you that to think of it only as the concentrations of the molecules determine the motions, that stops being true once everything starts moving.
Yes.
But before things move, it's okay to think that way.
Okay.
Other questions?
You get another chance also. In fact, you can interrupt also while I'm talking. You should remember that.
So before we go to coffee break, we're going to take the group picture now, here on this floor, below the stairs. So she's going. The person from the institute is going to take the picture from outside the stairs. So we all stay here now, right now. Okay? Thank you.